So welcome back. And in today's video, we are going to talk about the process of DNA replication. When we talk about DNA replication, we know that this process it does not take place randomly at any location, but at particular sites, and those specific sites are called as the origin of replication. Now, what are the prerequisites for the process of replication? First and foremost, we require is a template strand, then a primer which is attached to the template with a free 3' hydroxyl group. Now this 3' hydroxyl group, it will catalyze the nucleophilic attack to the incoming nucleotides. Like we know, we have a nucleotide and it is a deoxyribose with a base that may be a purine or a pyrimidine and a phosphate attached to it. Now this 3' hydroxyl group, it will catalyze its nucleophilic attack on the alpha phosphate with the release of the inorganic phosphate here. So this is how the incoming nucleotides they are added one by one. Now try to understand the process of replication step by step. We know that DNA that is a double helical structure. Okay, so in order to replicate, the first thing it needs is to unwind these two strands apart, and that step it is catalyzed by helicase enzyme. Now this helicase in presence of ATP it unwinds the two strands of the double helix. Once these strands are separated, these are stabilized by the binding of another protein that is the single strand binding proteins that is the SSBs. Now you can see that once this replication machinery as it proceeds ahead. So you can see here. So as the replication continues, so this machinery it will go in this direction. Now, as it proceeds, there will be a strain in the DNA molecule. Now we can see that from the same DNA molecule, you have two strands. Okay, so from the two strands which are acting as the parental strand, we have the replication of this strand as well as replication of this strand. So it synthesizes two DNA molecules from a single DNA molecule. Now here there will be a generation of the strain as it moves ahead. And this strand it is then removed by a topoisomerase by the break in the DNA, and then it basically passes the DNA through that break. So this topoisomerase here, it basically topoisomerase second, it helps to relieve the DNA from the strain or from that tension in the DNA molecule. So we understood the process. Now there has been a model proposed for the replication of the DNA. So as mentioned about the origin of replication, we say that this is the origin of replication. So in the origin of replication, we have 313 mers. So these are nothing but the repeats basically. So we have the 13 mer repeats, 313 mer repeats, and 5 9 mer repeats. And the process of replication it starts when a protein DNA which is bound to the ATP, it basically comes and it binds in this region, leading to the unwinding of in the 13 mer region as well. Now you can see that as it unwinds, then we have the DNA helicases. It will come and it will further lead to the separation of the two strands of the DNA. And this DNA helicase, it is aided by another helicase on another protein that is the DNA C. It is the helicase loader. Now you can see that once these two DNA helicase and DNA helicase loader, that is DNA B and DNA C, they come into action and they lead to the separation. So this is the replication bubble which is formed here. Now the next step is the primer. The primase enzyme it will come and it will synthesize the short primers, short nucleotides, oligonucleotides in fact, with the 3' hydroxyl ends, because we have earlier seen that how this 3' end it will catalyze the it will basically catalyze the addition of the new nucleotides, deoxyribonucleotides, so that this strand is extended. Okay, now what happens is after the synthesis of the primers on both the strands, both the leading as well as the lagging strands, this helicase is removed, and afterwards we have the polymerases, the DNA polymerase third holoenzyme, it will come and it will bind at this region on both of these strands. So remember, the first and foremost, the DNA polymerase alpha, it will come and it will carry out the synthesis. Okay, and then there is the replacement of this alpha by the polymerase epsilon and polymerase delta. Now these two enzymes, this is called as the polymerase switching. So first and foremost, DNA polymerase epsilon, it will carry out the synthesis of the leading strand, and polymerase delta that will catalyze the synthesis of the lagging strand. So that is basically how the replication proceeds. Now there is one important aspect That is, the E. coli DNA replication is regulated by the DNA A ATP levels and the seq-A protein. Now, how regulation happens in the process of replication in the E. coli, let's understand that. So as I mentioned it, we have these 13-mer repeats and 9-mer repeats. And we know that there are these palindromic sequences which are that is the GATC GATC. So these are the repeats which are present in the origin. And what is so special about these repeats is that these repeats are methylated. At the adenine position, you can see this red marks. This represents the methylation of these strands. So you can see both of these DNA strands, they are methylated. And once the replication initiates, so you see this replication bubble is formed and the new strand is formed. Now, as this new strand you can see here, but this new strand, it will not be methylated yet. Okay, so here only one strand is methylated, not the other one. Now, this hemimethylated state, which we call it, okay, this basically provides a landing platform or basically another protein that is a seq-A. It will come and it will bind to these hemimethylated sites. Now, what does this binding of seq-A, how it regulates the process of replication? What it does is that once the seq-A binds here in these regions, it will prevent the DAM methylase, that is basically which carries out the methylation on these adenines. So it will bind and it will inhibit the binding of the dimethylase and it will inhibit the binding of the DNA A. So you can see that when there is no binding of DNA A, there will be no replication. So it gives it more additional amount of time basically. So once one of the strand is replicated, there will be no reinitiation of the replication in that particular sense. Okay, so here you can see that once the CK is not there, it's removed, and the dimethylase it will come. Similarly, we have the DNA A, it will come and it will bind here. Once the DNA A binds, it will catalyze this. So this basically will prevent the immediate reinitiation of replication from the two newly synthesized daughter copies of the ORIC. That is the origin of replication. So